Hello, everyone. Today we're continuing our deep dive of Richard Dawkins and Yan Wong's book, The Ancestor's Tale. In this episode, we're going to discuss the migratory patterns of jellyfish. So let's jump right in. Dawkins and Wong estimated our common ancestor with the clade Protostomia at about 560 million years ago in the late Ediacaran period, and our common ancestor with Xenocoelomorpha, aka the acoelomate flatworms, if they are indeed sister to all other bilaterians, at about 565 million years ago. These dates are close to the recent study published in the journal Science. Carlisle et al. Used the relaxed molecular clock methods calibrated with fossil data to date our common ancestor with Protostomia between 564 and 577 million years ago, and our common ancestry with Xenocoelomorpha 569 to 582 million years ago. We talked about the confusion over the placement of acoelomate flatworms in the ragworm's tail, and in November 2024, a massive genomic study dropped that pinned the worms as sister to all other bilaterians, supporting the Nephrozoa hypothesis. Another fascinating recent study, this one from July 2024, reanalyzed a number of Pikaia specimens, finding it to be a stem chordate rather than a stem cephalochordate. But the study went further. It also found that the enigmatic Vetulicolians are not a single clade, but a paraphyletic assemblage of increasingly derived stem chordates. Contrary to our prediction in the aforementioned video, Banphia isn't a very primitive stem protostome, but a very primitive stem deuterostome. Vetulicolia is more derived than Banphia, Didazoan more derived still, then Yananazoan, Pikaia, and Cathaemyrus, and finally, the crown chordates. Today's tale brings us to 590 million years ago in the middle of the Ediacaran period, and now seems as good a time as any to tackle it. The Ediacaran was until very recently quite a mysterious time. The crown members of most modern bilaterian phyla appeared in the early Cambrian or late Precambrian. The so-called Cambrian explosion was itself a series of radiations of bilaterally symmetric animals, such as chordates, echinoderms, arthropods, onychophorans, priapulids, mollusks, and so on. But these animals must have come from somewhere. When we turn to the late Ediacaran, we see a variety of primitive bilaterians. We mentioned the recently discovered Uncus zaugizi in the last video, the nematoid worm. The recently discovered trace fossil Hematiicnus mangano that appears to have been made by a priapulid relative. There's Claudina, a primitive annelid. The enigmatic Elengia is either an annelid or panarthropod. Namacalathus is at least a spiralian and possibly a stem lophophorate, the group including brachiopods, bryozoans, and phoronids. Kimberella is most recently seen as a stem spiralian, and Icaria seems to be an early crown bilaterian. Remember that as we go backwards in time, discerning the affinities of fossils becomes increasingly difficult. A reminder how our modern classifications blur when dealing with organisms defining the basal clades themselves. That has been especially true for the animals of Ediacara Hills, South Australia. In the 1940s, Australian geologist Reginald Sprigg discovered a series of fossils he initially identified as a bunch of jellyfish. Sprigg was likely drawn to this conclusion by the earlier works of Charles Doolittle Walcott, the discoverer of the famous Middle Cambrian age Burgess Shale, as well as a few reports of possible jellyfish fossils in some Precambrian localities. None of these fossils, though, actually turned out to be jellyfish, and Sprigg's own fossil assignments were rejected upon further study. Others argued that the Ediacarans were octocorals, giant protozoans, or even terrestrial lichens. In more recent years, all of these assignments have been falsified, as the Ediacaran animals are not a single group at all. They are a polyphyletic assemblage of various clades, in their own way likely as diverse as anything seen in the ages since. For starters, the clade Proarticulata, including such members as Vindia, Dickinsonia, and Yorgia, appears to be a phylum of stem bilaterians. One of the most obviously animalian features of these organisms is their growth. As paleontologist Bruce Renegar says, quote, "As is well known, the body of Dickinsonia costata was constructed from segments, isomers, units, or modules, which appeared at an unresolvable size at the posterior end and continued to grow and change shape during the life of the animal. This alone is a feature of animals rather than plants, kelp, or fungi." Close quote. Another unique feature of this group is that many of its members possessed glide symmetry. In other words, each segment was slightly offset from the opposing segment. Interestingly, though, several Proarticulata do actually possess bilateral symmetry, such as Andiva ivancevi. These characteristics and combinations suggest a stem bilaterian affinity rather than sponge, ctenophore, placozoan, or cnidarian affinity. We will treat the bizarre trilobozoans, ernietomorphs, arboriomorphs, and rangiomorphs when we reach the sponge's tail. Once we leave the bilaterians behind, there is a fundamental change in how animal bodies are built. All this time, we've only met animals with three germ tissue layers: endoderm, mesoderm, and ectoderm. As developmental biologist Lewis Wolpert famously said, quote, "It is not birth, marriage, or death, but gastrulation, which is truly the most important time in your life." Close quote. Indeed, gastrulation is the period during embryonic development when you go from being a holoblastula with a two-cell thick layer called the epiblast and hypoblast to a gastrula with three germ tissues. The endoderm gives rise to much of the gastrointestinal tract, respiratory tract, endocrine organs, auditory system, and urinary system. The mesoderm gives rise to the notochord, cartilage, bone, kidneys, gonads, heart, blood vessels, blood cells, and the subcutaneous layers of the skin. And ectoderm gives rise to the skin glands, and neural tissue. All the other animals we're going to meet only have endoderm and ectoderm, no mesoderm. It should be unsurprising that homologous genes involved in patterning the mesoderm are known from the endoderm of cnidarians. Furthermore, it should also be unsurprising that transcriptomic analyses find endoderm genes are expressed first, then ectoderm, and lastly mesoderm, and the largest fraction of orthologs of choanoflagellate genes are expressed in endoderm tissue. This indicates that both ectoderm and mesoderm tissues were co-opted from endoderm. The next living clade we meet is complicated. In the Ancestor's tale, Dawkins and Wong treat the phylum Cnidaria first, which is followed by Ctenophora, the comb jellies, Placozoa, and then Porifera, the sponges. Cnidaria and Ctenophora were treated as a single clade called radiata, referring to their radial symmetry, for a long time by zoologists, but molecular genetics has strongly falsified this association. Instead, ctenophores and sponges spar for the designation of sister to all other animals, but that's a topic we'll pick up later in the sponge's tale. Cnidaria is routinely found to be sister to Bilateria, but where do the odd placozoans go? Placozoans are tiny, being about 3 millimeters long, amoeboid animals that lack most of the fundamental characteristics of animals more complex than sponges, such as a basal membrane, neurons, muscles, and a mouth. Instead of taking food into the mouth like all other non-sponge animals, placozoans engulf food with their ventral surface, which is covered with cilia enabling them to move. The first placozoan species, Trichoplax adherens, was named in 1883 and placed very low on the animal tree. However, subsequent studies argued instead that Trichoplax was either an adult flatworm or a juvenile cnidarian, being just the larval planula stage. Placozoans were rediscovered in 1966 and just a few years later were identified as a group separate from any known phyla again. However, the entire phylum still had just one species, but with hundreds of genetically distinct lineages. There are now 4 living species of placozoans: Trichoplax adherens, Polyplacatoma mediterranea, Cladterdia calaboinventa, and Hoelungia hongkongensis. There is also now one extinct giant placozoan species, being a whopping 5 millimeters in length, From the Triassic period, Maculocorpus microbialis. Fascinatingly, that fossil placozoan was found alongside the first ever fossil of a xenocoelomorph flatworm called Gutticorpus concentrica. Because placozoans are so simple, having even fewer cell types than sponges, some hypotheses have argued for their placement as sister to all other animals. This would affirm the simplicity of placozoans as indicative of their primitiveness. However, other hypotheses have found support for placozoans being sister to bilaterians, sister to cnidarians, and on their own with them splitting off before cnidarians. A 2022 paper by Michael Tesler et al. Found support for placozoans as splitting off before cnidarians, with cnidarians being sister to bilaterians. Also worth noting that this paper found support for sponges as sister to all other animals when morphological and genetic characters were considered together. So now we meet the phylum Cnidaria. Cnidarians are named for a character that they all share, the cnidocyte. This is the stinging cell, that jellyfish bit responsible for so many unhappy days at the beach. Within the cnidocyte is the cnidocyst, a bulb-shaped capsule containing a hollow coiled tubule tipped with a harpoon. Atop the cnidocyte is a hair-like trigger that, when activated, rapidly releases the coiled tubule, causing it to be injected in the skin of some animal. Many cnidarians are venomous, but the severity of their venom varies widely. Pacific sea nettles, Chrysaora fuscescens, will give you a mild itchy rash. The common kingslayer irukandji, Mallow-kingi, has killed people. And moon jellyfish, Aurelia aurita, cause basically no reaction at all. Many anemones are also non-venomous, which is why they feature so commonly in touch pools at aquaria. When one touches the oral arms of a giant green, plumose, painted, or aggregating anemone, one can detect a stickiness that adheres the arms to one's finger. Those are the cnidocytes embedding their tiny harpoons in your skin, but don't be alarmed, you're not in any danger. For such simple animals, cnidarians have a complicated life cycle, which itself may be a clue as to how long a history their lineage had before generating forms with their diagnostic cnidocytes. Like all other animals, cnidarians begin life as a fertilized egg cell, but this planktonic zygote develops into a benthic, pancake-like structure called the planula. The planula freely roams until it finds a spot conducive to growth, and then it develops into a tentacled polyp. From there, life can go in a number of directions. In corals and anemones, they can stay as the polyp for their whole lives. Hydrozoans may go through both the polyp and medusa phase or just either stage, and cubozoans and scyphozoans go through both stages. The medusa is formed from the polyp by a process called strobilation. Multiple individual young medusae, technically called ephyra, are stacked like cards that can float away with the current. Based on phylogenetic analyses of cnidaria, this indicates that the polyp was ancestrally the adult stage, with the medusa stage being built atop it. The medusa stage appears to have evolved in the ancestor of Medusazoa, but was lost in Staurozoa and Aplanulata, a clade of hydrozoans. Within cnidaria, the tree forks into the clade of corals and anemones on one side, and bizarre parasites, staurozoans, jellyfish, hydrozoans, and siphonophores on the other. We will treat the corals and anemones in the next tale, so let's stick with the other clade. Sister to staurozoans, jellyfish, and hydrozoans is a clade of very odd parasites called endocnidozoa. These have been very difficult to place historically due to their highly degenerate bodies, losing such things as their whole nervous system. One member, Hennegaia, even lost its mitochondrial genome, being unable to perform cellular respiration. Endocnidozoa contains two clades, Myxozoa, which contains about 2,200 species of minute endoparasites of invertebrates and vertebrates, and Polypodium, which infects the eggs of sturgeons and paddlefish. Endocnidozoa is sister to the clade Medusazoa, which is split into Hydrozoa, Staurozoa, Cubozoa, and Scyphozoa. There are some stem medusazoans, such as Aurora lumina from the late Ediacaran, 562 million years ago, and the order Conulatae, including Conularia, which extends from 545 to 205 million years ago, the latest Ediacaran to the end of the Triassic. Hydrozoa is a diverse clade containing individuals ranging from the tiny freshwater hydra, which lives as a polyp, to the giant siphonophore Praea dubia that can get up to 160 feet long. Hydra cells evidently do not senesce, or age. The reason is that Hydra stem cells continually self-renew, which is controlled by transcription factor forkhead box O, also known as FOXO. This transcription factor is shared with bilaterians as well, and experimental data shows that mutations in it affect animal longevity. Researchers have also shown that mutations in FOXO affect the functionality of Hydra's innate immunity. For vertebrates, researchers thought for a while that their cells, when properly cultured, could live indefinitely, but this was eventually shown to be false by anatomist Leonard Hayflick and cytogeneticist Paul Moorhead in 1961. The inability of normal human cells to live forever is now accordingly known as Hayflick's limit, and occurs because chromosome telomeres are not fully correctly copied during DNA replication. Within Hydrozoa is a clade called Siphonophore that has engendered much discussion about what it means to be an individual. The reason is that, like all other cnidarians, siphonophores start life as a zygote, grow into a planula, and then develop into a bunch of connected polyps and medusae, each called a zooid. If this is difficult to understand, try to imagine that these organisms always produce offspring as sets of genetically identical twins. Each of these twins is a zooid. Instead of going their separate ways, they will live together as if they are one organism, each zooid being specialized into a series of functions. Consider the siphonophore Nenomia bejuga. It has nectophores, which are zooids specialized for swimming, food-capturing gastrozoids and palpons, and then male or female gonodendrons, which produce sperm or eggs. It also has tissue called the pneumatophore that is thought to be filled with carbon monoxide to aid in buoyancy. This is what you see above the water of the Portuguese man-o'-war, Physalia physalia. The gastrozoids and palpons are homologous to polyps, while the nectophores and gonodendrons are homologous to medusae. A siphonophore, then, is just a giant floating polyp and medusa colony, similar to how a coral is typically a bunch of connected polyps. The difference is that coral polyps all perform identical functions, whereas the genetically identical zooids of a siphonophore are specialized into different roles. In that way, zooids are similar to organs, but zooids themselves are derived from formerly individual animals, much like how our mitochondria are bacteria. And just like mitochondria, each zooid cannot live independently. Nectophores cannot gather food for themselves, and gastrozoids cannot swim by themselves. Intriguingly, the hydrozoan family Porpidae independently evolved floating polyp bodies. The blue button jellyfish Porpita porpita is a small member that I've seen washed up on the shores of the Gulf of Mexico. It does look like just a little jellyfish, but like the siphonophores, its parts are specialized polyps. Dactylozoids protect the colony, gastrozoids gather food, and gonozoids produce the medusae. All the polyps in a porpidid individual are either male or female, so the medusae are either male or female too. The tiny male medusae produce sperm, and the females produce eggs. These gametes combine to form the zygote, which, like in the siphonophores, grows asexually into the floating polyp colony. The other genus in this family is Villela, also called a by-the-wind sailor. It looks a bit like the Portuguese man-o'-war, but with a long diagonal sail. Villela is preyed upon by pelagic gastropods such as the swimming slug Glaucus and the planktonic snail Janthena, who steal its cnidocytes for their own defense. Next, Staurozoa is often referred to as the stalked jellies because they have independently lost the medusa stage. Two staurozoans, Hautia and Memsedia, have been identified in late Ediacaran sediments dating to 560 million years ago. The next clade is Cubozoa, the box jellyfish, some of whom are the most venomous jellyfish in the world. One extremely bizarre feature of cubozoans is their eyes. We tend not to think of jellyfish as having eyes, but cubozoans do have eyes that are similar in many respects to vertebrate camera eyes. Remember back to the howler monkey's tail where we discussed how eyes work. Your retina is a layer of photoreceptors at the back of each eye that absorbs photons, generating a neurochemical message to be sent to your brain. Light passes through your cornea and is focused by your lens onto the retina, sharpening the image. This is broadly how vision works for all vertebrates. Eyes are outgrowths of the diencephalic region of the brain. First, the optic vesicle, or optic primordium, buds from the brain towards the surface ectoderm. The tip of the optic vesicle will eventually give rise to the retina, and the surface ectoderm in front of the optic vesicle is the lens placode. The retina invaginates first, followed by the lens placode. The ends of the lens placode eventually come together to close the lens. The cornea forms later in front of the lens. This broadly recapitulates the evolution of the eye. In mollusks, we can see every step of eye evolution from the flat photoreceptor sheet of limpets, to the cup eye of abalones, to the pinhole eye of nautiluses, to the primitive lensed eye of chitons and scallops, to the complex camera eye of squid and octopus. Richard Dawkins spends a whole chapter of Climbing Mount Improbable discussing eye evolution if you want a deeper dive. Turning to cnidarians, visual organs called ocelli have evolved in hydrozoans, cubozoans, and scyphozoans to varying degrees of complexity. To quote a 2002 paper by Vicky J. Martin, quote, "Simple eye-spot ocelli flush with the animal surface are demonstrated in Turris species and Leucartiara octona. Pigment cup ocelli are found in Polyorchis penicillatus, Spirocodon saltatrix, and Bougainvillea principis, and pigment cup ocelli with a lens are observed in Cladonema radiatum and Sarsia tubulosa. The most complex Hydromedusan ocellus is an inverted type found in Tieropsis multiserrata. This ocellus has a gastrodermal pigment cup separated from the photoreceptor cells by a layer of mesoglea. The tips of the photoreceptors project into the lumen of the cup, while their basal ends pass out of the opening of the cup. The ocelli of hydrozoans are found at the base of the tentacles. Meanwhile, in cubozoans and scyphozoans, the ocelli are associated with a statocyst, which are regions that sense gravity. And the whole organ is called a rhopalium. Rhopalia are found on the medusa at small indentations on the fringe of the bell. The most sophisticated cnidarian visual system exists in cubozoans. On each of the four corners of the medusa, the rhopalia has 6 visual organs. According to a 2020 paper by Kristen M. Koenig and Jeffrey M. Gross, quote, 2 eyes in each corner have a lens, the others are simple pigmented cup eyes. The 2 complex eyes have a cornea, iris, lens, retina, and pigmented layer. The upper eye is used for navigating mangrove forests of the cnidarian habitat, and the lower eye is used for viewing the underwater environment, close quote. Yes, you heard that correctly. Cubozoans look upwards at the mangrove trees which hang over the water. This bizarre fact brings us now to the jellyfish's tale. During World War II, sonar operators were puzzled by what seemed to be a false bottom of the ocean that rose in the evening and sank in the morning. This false bottom is called the deep scattering layer, and though naturalist Georges Cuvier noted that the water flea Daphnia underwent a daily vertical pattern as long ago as 1817, nobody knew just how widespread this phenomenon is. The oceans are vertically stratified into zones of varying sunlight. Closest to the surface is the most sunlight, and descending means less sunlight. The areas of sunlight, called the photic zone, are full of phytoplankton, which are photosynthetic cyanobacteria, foraminifera, radiolarians, coccolithophores, diatoms, and other types of algae. These act analogously to the grass of the savanna, and analogous to zebras and gazelles are numerous crustaceans, such as copepods and krill. But these don't just graze the phytoplankton at any hour of the day. Rather, they wait until night. At night, predators are fewer in number, so these tiny crustaceans ascend to their food. Following behind them are larger predators: lanternfish, the world's most abundant vertebrate, squid, siphonophores, and dapper dinosaurs' favorite animals, the pelagic tunicates. These also vertically migrate in uncountably large numbers. If phytoplankton are the grass and krill are the zebras, then jellyfish are one of the big cats. This is where the analogy breaks down. Jellyfish, unlike lions or cheetahs, do not pursue individual prey items. Rather, their tentacles drift passively behind them as they swim, catching whatever is unlucky enough to run into them. On the island Mercershar in the Pacific, another type of jellyfish migration takes place in the so-called Jellyfish Lake. This lake is connected to the ocean and is thus saltwater, supporting an enormous population of jellyfish. Though moon jellyfish are found there, the dominant species is the golden jellyfish, Mastigius papua eptisoni. These jellyfish are positively phototactic, meaning they swim towards light. In the morning, the population of some 7 million individuals swims towards the eastern end of the lake, and as the day progresses, they swim back to the western end. As it happens, the golden jellyfish don't run into the shore because trees ring the lake, casting shade. When a jellyfish winds up under the shade of a tree, it swims back to the light. Why do they do this? The leading hypothesis is that the jellyfish's endosymbiotic zooxanthellae, more on them in the next video, require lots of sunlight for photosynthesis, so the jellyfish track the sunlight. And that's the jellyfish's tale. Cnidarians have been around for some 600 million years and diversified into forms as disparate as corals, siphonophores, and jellyfish. Independently of bilaterians, jellyfish and their kin have developed their own visual systems for tracking light and prey, which is one of the features that has contributed to their immense success. In the next video, we'll look at some important adaptations of corals, endosymbiotic algae, and a tough skeleton. So, thanks for watching, and we'll see you all next time.